Reproduction is one of the most important characteristics of life because all life has the ability to make copies of itself. And since life is made up of cells, cell division is at the core of this process. It's the process by which mother cells make copies of themselves or daughter cells. And all life has to do this because life must grow and develop. Organisms must repair and replace old parts. And also, living things need to reproduce. And for some things, cell division is the whole of the reproductive effort. Because for unicellular organisms, all they have to do is basically make new copies of itself. But all organisms must do cell division because cells have a limit to their size. When you look at a large organism, you're not actually seeing an organism that has large cells. What you're actually seeing is an organism with many cells. And that goes back to the idea that folding equals efficiency in life. Because think about cell size as an example. As the cells grow, the cell membrane, which represents the surface area of the cell, actually grows two-dimensionally, while the volume or the cytoplasm of the cell grows in 3 dimensions. So it gets to a point where the surface area to volume ratio will be too much for the cell to actually deal with, because the cell membrane will not be able to efficiently exchange materials with the environment, bringing in the nutrients and putting out the cell products and wastes that the cell wants to get out of it. So basically, cell size limitations, growth, replacement, and reproduction are reasons for which the cells must divide. The cells do this in 2 ways. They do it through a simple process called binary fission, or more complex cells do mitosis or meiosis. The simple cells, like prokaryotes, such as bacteria and archaeobacteria, all they have to do is basically split up after making a copy of their DNA. Their DNA is a circular chromosome that is a continuous molecule that is very small compared to eukaryotic DNA, and they don't need to do anything special to replicate the cell. They just basically take a copy of the DNA and they cleave the cell in half, making sure that each of the nucleoid regions where the DNA is are separate from each other so that the DNA will be in separate cells. But the more complex cells that have internal membranes, including a nucleus, have to deal with the nucleus separately. And they also have these complex chromosomes, and many of them. And these chromosomes are linear molecules which are packed up around proteins into a scaffolding structure that's very complex. And they have centromeres to connect each of the copies of the chromosomes called sister chromatids. And each sister chromatid has a long arm called Q arm and a short arm called petite arm or P arm. And these are all held together by the centromere. And then that centromere will separate in the middle of the copy process. But until it does, it will stay together, making this familiar X-shaped chromosome, which is visible only during the division process. Cells, like eukaryotic cells, will usually have multiple of these chromosomes, and that's why it needs to have a specialized process to separate them equally and efficiently. And they also have to deal with the nuclear membrane and all the other structures inside the cell during the cell division process. So mitosis will be a much more complicated process of leading up to cell division, because mitosis is not actually cell division. That's cytokinesis in eukaryotes. Mitosis is really nuclear division, or karyokinesis. And by the way, eukaryotic cells do a special kind of cell division when they want to create gametes or haploid cells, and that's meiosis, and we'll talk about that on a future review video. Mitosis happens as part of a larger cell cycle. It starts after a division with G1 phase, where the cells is growing because they're smaller than usual because they just separated, they just became daughter cells, and they will replicate all of their organelles and start making protein synthesis to grow up, and they will also continue their normal metabolic activities. They will be doing cellular respiration. If it has chloroplast, chlorophyll will be doing photosynthesis. They'll be doing membrane transport, protein synthesis. They'll be doing all of the activities that the cells normally do. And they'll be growing up to a normal cell size. When you get to the S phase, the DNA will be doubled. So during the S phase, the DNA is not doubled yet, but it's doubling as you go through the S phase. And it's going to continue doing everything it did during the G1 phase, but add a layer to that where the DNA is actually being copied. During the G2 phase, the cell already has doubled the DNA since it already underwent the S phase. Now the cell is getting ready for division, and it will get to the point that it will start replicating, doubling its organelles, and it will start becoming extra large size, so that when the cell actually splits in half, it won't be too small to begin with, and then it will finally go into the mitotic stages, and then at the very end of that, quite at the same time that the last mitotic stage is happening, cytokinesis will happen, and the cells will actually split up. And that's the cycle that cells normally undergo. By the way, the large G1S and G2 phase, which take as much as 90 to 95% of the cell cycle, depending on the cell, is basically called the interphase. During the interphase, cells continue to make its normal activities. They do all the metabolic things that life does. But during mitosis, they stop doing all of that and focus on cell division, which is why some cells in body organisms will not undergo mitosis ever. After they start working, they will stay in the interphase forever. We call those G0, and those G0 cells will not undergo mitosis because they cannot afford to stop the metabolic activities. Think about some muscle cells, some fat cells, and some nerve cells, which will be constantly doing the job that they need to do, and so they can't stop doing what they do. Nerve cells, another reason why they can't split up is that who you are is basically the connections that exist between nerve cells, and to split the cell would mean to break those connections, and which will erase all your memories. And so nerve cells can't actually ever split. And that's why these tissues have trouble repairing themselves after they actually become active and reach the point of completion of the tissue. So after adolescence, you're only losing nerve cells, you can't actually gain them, with very few exceptions. Let's talk about mitosis in a little more detail. But before we do so, remember that before every cell division, you're always going to double the DNA. Chromosomes are actually linear molecules, one continuous linear molecule. But during the cell division, by the time you get there, you already underwent the S phase, which is when the cell doubles itself. Remember, everybody has to undergo the S phase. Even prokaryotes will double its DNA before division. Because if you're going to make two daughter cells that are going to be identical to each other, each of them have to have all the blueprint or the entire master plan, that's the DNA. And in eukaryotic cells, that happens by getting that one linear chromosome and making another copy of it, and keeping it attached until it's time for it to efficiently split in an organized way. And that's when you see that X-shaped chromosome that's familiar. You will see that only after duplication actually happens during the S phase, and after the chromosomes cover themselves up at the beginning of mitosis, during prophase. And eventually, each of those copies will split themselves and go into their different daughter cells. And so here we have the process of actual mitosis. Some of the things you need to know about. Spindle is what it looks like when you get to metaphase and you see this whole structure all put together to make the cells separate efficiently. Kinetochore is the actual region right next to the chromosome where the spindle fibers will connect to actually help pull the chromosome. Fibers are those parts of the spindle which are actually lining up from each pole of the cell all the way to the middle to help drag the chromosomes to the extremes. You have the nuclear envelope and the nucleolus, which are the first things to disappear. If you see a nucleolus in a cell, the cell is going to be in interphase. Nucleolus is only needed when ribosomes are being made, and ribosomes are only made when protein synthesis is being made. So if interphase is active, you have nucleoluses. But if the cell is no longer performing its normal metabolic activities, you no longer need the nucleolus. And so that's one of the first things to disappear. And then the nuclear envelope disappears because you're going to need to start organizing these chromosomes in different ways. All of this will happen during the prophase. At the end of prophase, the chromosomes are already coiled up and the spindle will start to form. And at that point, we call it the prometaphase, when the kinetochore fibers attach themselves to the chromosomes, and the chromosomes start becoming lined up towards the equator of the cell, and that's when you get to metaphase. Now, let's think about it. Why go through the process of actually having prophase? Why package the chromosomes in the first place? Well, one of the reasons why chromosomes exist is to preserve the DNA code in an actual organized structure to facilitate cell division. Imagine dragging the chromatin, which is what it looks like during interphase. It will be too messy to actually try to separate that exactly the same in each daughter cell, especially when you consider that the DNA will be doubled at that point. And so it will be too much DNA to manage. That's why we have chromosomes. We also have chromosomes for other reasons, and we will revisit that when we do chromosome inheritance later in the year. Also, why would you go through the metaphase plate? Why line up the chromosomes in the middle like that? What would you gain from that? Why can't you just, as soon as you have the chromosomes formed during prophase, just start separating them to different ends of the cell? Well, that would make that sometime you might send a chromosome to one side instead of another and actually have an unbalanced amount of DNA per daughter cell. Having the chromosomes line up and having the sister chromatids face opposite ends of the cell and then cleave the centromere and drag them to opposite ends of the cell guarantees that each daughter cell will have exactly half of the doubled copies. In other words, each of them will have a complete copy of the DNA code. Now, the chromatids will then be each chromosome or one complete DNA information from that chromosome. So with the chromosomes you see as axes are actually two copies of that. Also notice that animal cells will have centrioles, which are a special pair of organelles that comes from a specialization of the cytoskeleton. They're made of microtubules, which are the same things that the cytoskeleton is made of, and they will facilitate the formation of the spindle during the prophase and metaphase period. And they will be connected to the edges of the polar end of the cell through aster fibers, which will help drag them to position and create tension to actually help split the spindle fibers. Also, notice that spindle fibers shorten during anaphase to actually make the chromosomes go to the corners. It's not like an elastic that's snapping and making the chromosomes go to the corners. Enzymes actually will destroy spindle fibers little by little and causing the spindle fibers to actually be dragged to the corners. And experiments have been done where you actually cut the spindle fibers or prevent the formation of spindle fibers near the poles, and it will still shorten and still go to the opposite ends. They just won't reach the poles if that happens. And so it's during anaphase, through the action of enzymes, that the centromeres are actually cleaved, and through the action of a different set of enzymes that the spindle fibers are shortened and that the chromosomes will head towards the polar regions of the cell. Remember that enzymes are involved in all of this. In fact, the nucleolus destruction and also the nuclear envelope destruction that happened during prophase would not happen if it wasn't for another set of specific enzymes. If you don't have these enzymes, these things don't happen and the cell cycle will be arrested at that point. Speaking of getting to the poles, when the chromosomes reach the ends, you're going to have to get to telophase, where the chromosomes will start to uncoil, the nuclear envelope will actually rebuild itself with the action of enzymes, and nucleolus will eventually come back. And in the process, that's pretty much the reverse of what happens during prophase. And at the same time, the centrioles, if it's an animal cell, will start to disappear, and the spindle fibers are going to be destroyed since they're no longer necessary, and the cell will also start the process of cytokinesis almost at the same time that all of this is happening. And this is basically the process of mitosis. What's important to understand is that this actually happens fluidly. There's no separate stages. This all happens as one continuous picture. And watching a video or an animation of this will really get into your head. This is just one big thing that's happening continuously. There's no stages. This is just how we actually study or divide them to make logical sense for us. Now, cells actually divide at the end of mitosis almost at the same time a telophase is happening in a process that's called cytokinesis, which will be another difference between plant and animal cells. Remember, the first difference was the presence of centrioles. The second difference is that since animal cells don't have cell walls, they just actually fold inwards, kind of similarly to the way that some bacteria and other unicellular organisms do it, and it will get their cell membranes and fold their cleavage inwards and actually separate the daughter cells. And that's how cytokinesis happens in animal cells. In plant cells, because they have the cell wall, if you have a room, the easiest way to separate the room is not to collapse the walls, but to build a new wall in between. And so that's kind of how plant cells do it with something that's called the cell plate. And that's yet another way how plant cells and animal cells are different. Also remember that both cells will have something that's called centrosomes, which will facilitate the process of formation of spindle fibers, which in both cases are made of microtubules. It's problems during mitosis that lead cells to actually become cancer cells. Cancer cells, by definition, are cells which are undergoing crazy mitosis. Instead of spending only 10% of the life cycle in the mitosis stage, these cells spend the majority of their life cycle in mitosis. They go through interphase very quickly and quickly start reproducing again. And these are cells which lost control of the cell cycle, and, for example, they do not respond to cell signaling the same way other cells do. Normal cells, when they get close to each other, they actually stop copying. They feel it's something that's called contact inhibition. When they touch each other, they stop growing. But in terms of cancer cells, they do not have contact inhibition, and they will start piling on top of each other and keep growing until they actually form a tumor. And this is actually something that we discovered in the lab when we realized that cancer cells would actually start growing even past the point where they touch each other. And speaking about the loss of cell cycle control, all of this process is controlled by chemical messengers inside the cells. Cells actually have what we call gateways before they enter each stage of the cell cycle. The first gateway is the G1 checkpoint, where the cell decides, if I'm big enough and I have a favorable environment here, then I should start copying my DNA because I've started to think about becoming too big and I have to copy myself. And this basically happens by the accumulation of something that's called cyclins. And these cyclins, or CDKs for abbreviated, will accumulate inside the cell as the cell grows up, and when you have enough of them, that's a message that it's time to actually split up. Then the cell goes into the S stage, and the DNA will be copied until you get to the point where all the DNA is copied. If the environment is still favorable and the cell's definitely enough, after a growth of G2 phase, enough cyclins will be in there that they will actually initiate the signal for mitosis, what is called mitotic CDKs or MPFs, mitosis proliferation factors. And when enough of these gather up, you pass the gate of the mitosis stage, and then we'll continue going in that stage until you get to the metaphase, and where another gateway will be there to determine, are the chromosomes all ready? Are they all called up? Is the spindle ready? Are they all lined up in the middle? If they are, the last gateway gets through and anaphase actually completes, and the cell undergoes towards the G1 cycle again. And all of this is controlled chemically inside the cell. I hope this all makes sense to you and you learn a lot about mitosis, the important process of cell division tied into a lot of the basic functions of life. Alright? Don't do anything that would make a mama proud.